So here we will discuss about the complement system. The complement system. So basically why this is called as complement system means there are a set of glycoproteins which help the antibodies in clearing or in responding in destroying the antigen. So they will complement the activity of antibodies, and hence all these group of glycoproteins are considered under the complement system. These complement components, they are glycoproteins, and these are basically produced by liver hepatocytes, mostly by liver hepatocytes. Also they are produced by blood monocytes, macrophages present in the tissues, and the epithelial cells present in gastrointestinal tract and genitourinary tract. These complement components, they are actually present in the serum. They circulate in the serum as proenzymes or zymogens. These are the inactive forms of the complement system. These are the inactive forms because they actually possess an inhibitory site. This inhibitory site has to be cleaved away. This has to be cleaved by proteolytic enzymes so that the complement components become active. These complement components, they are generally designated by numericals or by letters or by trivial names. Numericals, like we have complement components from C1 to C9, a series of complement components. For example of letter designations is like factor D, factor B, we call them like that. They are also named in trivial names like homologous restriction factor. These are just the examples to tell you how these complement components are basically designated. And coming to an example of these complement components, these complement components usually they undergo cleavage. When they undergo cleavage, they are cleaved into one large segment, and usually this large segment is the one which goes and binds to the target whichever has activated this complement. And other component, that is smaller fragment, the small fragment will diffuse away, and it is responsible for localized inflammation. Let us take the example of one complement component, say for example C3. C3 can undergo cleavage into a larger fragment, and this larger fragment is now designated as C3b. Okay, similarly the smaller fragment which is derived is now named as C3a. This is about the cleavage of different complement components. So this forms the basis for understanding of different complement pathways. This complement is activated, and it will function in three different pathways. One is classical pathway, the second is alternate pathway, the third is lectin pathway. However, in the syllabus we have classical pathway and the alternate pathway only. Before talking more details about the classical pathway and alternate pathway, let us first talk about the functions of this complement. The functions of complements were of four important aspects. One is lysis of the target, second is opsonization, third is inflammation reaction, and the fourth is removal of immune complexes. Let us observe the pictures to have more details about it. So you can find a target cell here. This can be any antigenic cell, maybe bacterial cell or any other cell in which the antigen is present. So this is acted upon by the complement system, and the target cell is lysed. The second function is opsonization. In opsonization, you can see here the bacterial cell is first coated by the complement, and now the phagocytic cell will have the receptors for the complement, so that the complement which has bound to the antigen can now bind to the phagocytic cell, and phagocytic cell will do phagocytosis. In this way, destruction of antigen is done. The third function is activation of inflammatory response. So this is a mast cell. On the mast cell there are receptors for complement binding. So the complement which has bound to the antigen, or simply the complement can bind to the mast cell, and it can cause degranulation. We know that the granular contents that are released by the mast cell are responsible for inflammatory response. So in this way, complement can do its third function, that is activation of inflammatory response. The fourth important function of the complement is clearance of immune complexes. So we know that basically the antigen shown in blue color round shaped here, so it is bound by the antibodies, and now whenever the antibodies are bound to the antigens, the antibody exposes a specific site on its Fc fragment, which where the complement can bind. So this we should understand. Simply antibody, when it is not bound to the antigen, cannot bind complement. Only antibodies that are bound to the antigen, they undergo conformational changes such that in their Fc region they expose a site where the complement can bind. So in this way we understand that the complement can bind only to antigen antibody complex. So further, these complements, as we have already discussed, there are receptors for complement binding on phagocytic cells. So in this way, complement they bind to antigen antibody complex, the immune complexes, and they will allow these immune complexes to bind to phagocytes, and engulfment of these immune complexes, clearance of immune complexes by phagocytes is also helped out by complement in this way. So complement system has got four major functions. One is lysis of the target cell, second is opsonization, third is activation of inflammatory response, and the fourth one is clearance of immune complex.